Welcome everyone. Welcome to this class. In this class, we are going to discuss about viruses and future. Actually, we are going to discuss how these viruses can help us in the future or in upcoming time. Okay. Now, in the 18th century, when people get to know a little about viruses, then the definition of viruses came out to be full on negative definition. Okay. In those days, people consider these viruses. They don't know exactly that these are virus are particles. Okay. So they consider them to be like some fluid or some material which are totally invisible. We can't visualize them, we can't filtrate them, and they always cause pandemic, epidemic, etc. But later, when Ivanovsky discovered this virus, right, so its image got improved. Okay, now after this improved image, although these viruses are known to be one of the deadliest microorganism on the planet, which cause epidemics and pandemics like COVID-19, as you have seen in recent years, later studies shows that they have potential. How they are potential? They are potential in order to help and improve human life. Right. So here we are going to discuss about the application of viruses, how they are helpful to us humans. Okay. Now the first point is the point of gene therapy and gene silencing. Okay. Now in gene therapy, I have explained you about the gene therapy, how we incorporate a useful gene into a virus and that is transferred to host. Okay, so it can be used to cure diseases like sickle cell anemia, hemophilia, rheumatoid arthritis, cystic fibrosis, and severe combined immunodeficiency syndrome. And this severe combined immunodeficiency syndrome is quite severe actually, right? And similarly, it can be used in gene silencing also, which is virus mediated gene silencing. Okay, so it is used to suppress the expression of genes in plants. We are not using this gene silencing technique on humans because it can cause various effects on us. We are basically experimenting on plants only these days, but we can do on humans also in later stages if it is considered safe. The next point is biopesticides, and as you know, the viruses are very specific in nature. They only target a certain insect, in this case, in the case of biopesticide. So let us say we have viruses like baculoviruses, nucleopolyhedrosis virus, echoviruses, lepidoptera family. Okay, now these viruses are those viruses those only affect insects, and we can specifically select those viruses which affect the certain type of uh, you know bugs or insects. Let us say there is a one crop. I have this crop right here, and this is suppose a bug. So we can choose that particular virus which will affect only this bug, so that this will not affect our crop in the later stages. That's how we use them as biopesticides. Now the next point is vaccine development, and you have also known these days how vaccines are important, like COVID-19 vaccine we have for Pfizer, and Indian that biotechnological vaccines we have also created. Like there are attenuated vaccines. Okay, attenuated in the sense ki you know they decrease the potential of virus to cause infection, and they basically make them a little, you can say, half dead. Okay, so that whenever they are used or injected into the blood as a vaccine, they will stimulate our immunity, but they will not cause an infection. Okay, then comes subunit vaccines. Now these subunit vaccines have proteins, a part of protein which can be found in the viruses, right? So we can attach them to those viruses which are not affecting us or which don't cause an infection to us, and send them into our system so that immunity can create memory cells, so that in future if that viruses attack us, we already have immunity, or at least we remember what these viruses look like. Then comes virus like particles. These VLPs, virus like particles, they are, you can say, they are like capsid part of the virus, or maybe alone nucleotide. They are not nucleotide, let us ignore the nucleotide for this time, only capsid. Okay, so they have potential to become vaccine. How? Because you can just think of them as a shell. Okay, in that shell, if we inject those shells to the body, then they can be used in various ways, like to induce immunity, to deliver drug, and we will discuss this deliver drug right now, which is the fourth part, as a bio nanoparticle or drug delivery particles. Okay, now these viruses have, first of all, they have large surface area. Okay, the smaller the thing, the larger their surface area gets. Okay, and they have multivalent targeting unit. Now these multivalent targeting unit are this A, B, C, and D part here. These are multivalent targeting unit. Okay, now these will multivalent in the sense they have multiple sites which can attach to animal cells. Okay, so let us say animal cell have A and C complementary, so A and C site can attach to this animal cell, and let us say another one have B complementary, so this B site can attach to that also. So this is very helpful, like it will not miss any cell and can cure as well. Okay, and the other thing is drug delivery. So in drug delivery, like we use its capsule. This is the capsule, I have highlighted it, and in the center it is the drug part. So we can enclose that drug part into the capsule, and that can also enter, can be, sent into our body or system in the sense is our body also. Okay, and it can deliver the drug. And first of all, it will avoid toxicity, because the size of virus is too small, so the drug which we send using virus, okay, it will be in very small quantity, and due to its small quantity its solubility will increase, right? Just you can say like a finer particle of sugars dissolve in water more readily compared to the those which are a little big. Okay, so that's how virus will also work, and the drug will be more soluble. And the last point is the bacteriophage therapy, right? So here is the bacteriophage, it will only attack on bacteria, so that's why we can use these phages to attack viruses, right? So it can be helpful against bacterial infection and also against multiple drug resistant bacteria. Now in these days antibiotics are not working against bacteria, that is because these bacteria are becoming resistant to drugs, right? But they cannot become resistant to viruses because they are their natural predators. Not exactly predators, but they attack them. So that's how we can use these bacteriophages to attack bacteria, and even these MDR problem, multiple drug resistant problems, can be cured. And that's it for this session. Thank you very much and see you soon.